Okay. All right, everyone, we're going to get started.
Wrong date there.
Did I pick the wrong one? No, it's the right one. Okay, so how many things are wrong with this slide? It's not February 21st, and that's not the CME code, so don't worry. Today's CME code?
Yes.
All right, so now we're in the right slides. This is today's CME code. Going forward, where's Ryan? He nominates the most people. But for the All-Stars, send the email nomination to Tico while we're finding Will's replacement, Tico, who is himself an All-Star. I don't think he's ever got, we'll give him an All-Star if he does a good job in the transition. Just send your nominations to Tico. Important, our department's promotion extravaganza is Tuesday, March 19th at 5 to 7 in the Bryant Building. Joel's going to talk a little bit about promotions today, but there's dinner served. We did it 2 years ago, and it was a big success, and so we're bringing it back again on the 19th. So put that in your calendars. This is the memory group. This is something Kim Johnson organized, this thing called the Wreck-It Rage Room, where you just go in there and they give you sledgehammers and you beat up on dishwashers and plates and glasses and all kinds of things. It was good. We had the whole memory group plus a big chunk of the folks who work with us at Maureen Road, and it was a great time. And thanks to everyone who was there. Duke was nominated for the best, not nominated, elected the best hospital in North Carolina. Congrats. This week's All-Star is Natalie Dupay, nominated by NADA for help with a very difficult patient. So she'll be getting a coffee cup. Research Roundup, Deb Kotite, Joel, Mike Lutz, and former Victoria Stabile, who used to work in the department, were authors in an article in Clinical Neuropsychologist. Get this, that looked at old doctors to set up normative cognitive profiling with the false hope that you could use this to encourage people to retire at certain points in their career, but it'll never happen ever. So this is just a fantasy paper that will never ever be actually used. So I don't know how many of you, Joel, did you volunteer for this?
No,
I'm not. And I'm not cognitively intact enough, so I didn't volunteer either. Yeah, but I've seen several physicians like this, and even if they're demented, you have to make reasonable accommodations. Do you believe that? That the doctor you see might be demented, but the law is saying you've got to make reasonable accommodations for that? So, yeah, I found that hard to believe, but it is in fact true. Nada El-Hosseini was a first author on a scientific report article looking at retinal arteries and cognitive decline in people with vascular disease. And Katie Peters, right, Raj? Remember Katie Peters? She was at Hopkins with us, don't you remember? Maybe you had left by that point. Yeah, me too. Looked at anti-seizure medication discontinuation in patients with brain tumors. I thought I was reading this morning. The question is, what is this guy's motivation? None of us have got, best I can tell, got our COVID vaccine this year. I won't ask for a show of hands, but I have, and nobody's really gotten it. This guy got 217 COVID vaccinations over the course of about a year, getting one per day in the month of January, alternating arms. It's not clear if he was doing this because he had obsessive compulsive disorder or he got one of those vaccine cards and was selling them on the open market, but good for him. Good for him. Now we're going to do Educate the Educator. Joel's going to give us a talk, and this is again part of the promotion series. And thank you, Joel.
Good morning, everybody. So when you talk about promotion, you're usually thinking about faculty members, but this is also meant to be kind of a heads up to the residents and fellows, because believe it or not, the clock has started to tick. The race has begun. So this is all about being organized with a lot of the things that you do as an educator. And this is not only for clinician educators. On the APT committee, when we look at promotions, we look at all the things people do. We look at all the educational endeavors that one takes. And really what I'm striving to get you to take ownership of is to create an educational portfolio for yourself. This is really important. You know, it's really easy to slip the posters you do and the papers you write into your CV, but there are other things you need to keep track of. And it's really important to demonstrate not only quality, but quantity. Quantity matters. You might not think it's an important contribution you made, but you should leave that to others to decide. And you really want to try to develop a local, regional, and national reputation if you truly want to get promoted on a clinician educator track. And the lesson is just keep track of everything and start now. Figure out where you're going to put it. Like in my day, we stuck it in a file in the drawer. In your day, you stick it in a file in your computer. These are all the talks you gave as part of any lecture series. These are articles you may have written. These are curricula you might have put together for any kind of educational program. And publications, including non-peer-reviewed publications. It all counts. We do a lot of mentoring all the time, and we don't always keep track of it. So if you have a longitudinal relationship with somebody, keep track of who you helped, when and where you helped them, and put that in your educational portfolio. Look at your local department and institutional efforts. Perhaps, you know, if you're a faculty member and you join Katie Moore's Educator group and you participate in that regularly, perfect. Perhaps you're Elijah running programs to teach people about how to use different databases. Perfect. Perhaps there's an institutional effort. You're a house officer and you're working on a GME committee. Put that in your educational portfolio and get involved in your medical and neurological societies. That can be nationally, like at the AAN. You know, to be on a committee for the AAN, basically it's as complicated as you going on the website, looking at the committee section. There's a little thing there that says, I want to apply to be on this committee, and you send an email to somebody. It's really not complicated. And they're looking for folks. They love young faculty. They love residents and fellows. You know, you just have to take the effort to do it, and you make great relationships there, and you learn a lot about how things work. Also, your subspecialty society. Perhaps you go to the epilepsy meeting. There's all kinds of subcommittees and all our subspecialty societies that you can get involved with, and it's really fun. And you meet people from outside of Duke and develop relationships that will help you in your career. This networking is the key to getting some activities outside of Duke, which is part of the key to get promoted. You could be the greatest thing on water at Duke, but it really helps when you're trying to get promoted to have relationships outside of Duke, to be doing things outside of Duke for the greater good. Maybe some of your colleagues decide to do some clinical research in education, and they say, hey, will you do this with me? And now you have something you can put in your publications. All publications matter. It's not just funded research, and you can get educational research funded, but it's unfunded research. So if you're going to change some curriculum, think about, well, maybe I could do a pre-test of the people I'm changing the curriculum for, and then do a post-test and prove that I changed some knowledge base and write it up. And what's really cool is that now there are journals that are friendly to educators. We had Roy Stroud here, and Roy's really advocating neurology education. So if you look at that journal regularly, you'll see studies about different changes in educational practice, all kinds of things that you could potentially pursue if you're interested in being a clinician educator. Academic medicine's another good place to publish. And do posters. There's an educational poster session at the Academy. You know, you can do a poster for it and then submit it for publication. So you have to kind of put yourself out there. You know, when people show up and say, oh, I want to be a clinician educator, it's kind of like saying, I want to be a basic researcher next thing. Like, okay, show me what you've done. And that's where you need to know what part of your story is, to not only get a job as a clinician educator, but also to get promoted as a clinician educator. And develop teams that you can work on. So once again, there are a lot of people at Duke interested in education, and you know, a lot of us know who those people are. If you're interested, you know, talk to Suma, talk to Katie, talk to me. We're all happy to help you find a path. And there are people that are specifically career educators at every major institution that you can link up with, that can help you with projects, that can advise you, that can tell you what's available at your own institution outside of your own department. And, you know, if you're doing a research study, also involve a learner, because not only do you get help doing the study, but you get to mentor somebody. They get a publication out of it too, which they need. And it all kind of feeds the cycle. This is a good reference if anybody wanted to grab a reference to look at clinician educators. I'm happy to send it to people after the talk, but we're all doing this. My plea to you is start keeping track of what you're doing. Put it somewhere, everything. Even if you think it's minor, you can always edit it out later if somebody helps you, you know, on your education portfolio for submission. Somebody can help you edit it out. But if you don't keep track of it, you're not going to edit it in. Okay, thanks a lot.
Thank you, Joel. That's tremendous advice. And I do want to remind you all that you heard that advice from this year's recipient of the A.B. Baker Lifetime Teaching Award from the American Academy of Neurology. So, well deserved, Joel. Okay, here's the CME again. Okay. I think I'm still sharing, right? All right, so today, again, another typo. Today is Raj Ratan is giving grand rounds. We're going to have Wayne introduce him, but I just want to introduce the Janice Massey lectureship, which Raj is giving today. We call it the Leaders in Neurology series. We all know Janice, and Wayne is on the line, and I'm going to ask him just to make a comment. So all of you know about Janice's academic accomplishments. What you may not know about Janice is that she started her career as a high school math teacher after graduating from Abilene Christian University. She then spent her time teaching, supporting Wayne, who was going to medical school at Georgetown, I believe. And then famously, she came in upon Wayne and his friends studying and said to herself, these guys aren't so smart. I can do this. And the rest is history. Wayne, do you want to make a comment on that scene? Were you there? Is it apocryphal? Is it true? What happened?
Absolutely true. And we often would study together, often in our living room, and she'd come in, listen. And of course, in those days, women didn't grow up going into medicine. It's quite different. And so she hadn't thought about it until she realized she could do it and wanted to do it. So it's a combination of both.
Thank you so much. Thanks, Ray. It's an amazing story. And yeah, and Wayne's amazing too. So these are the past recipients. Raj knows a bunch of them. And it's an all-star group of people, and Raj is, Raj has made just every bit as important contributions as all these folks. There's very little advantage to getting old. First of all, they ask you to be involved in cognitive studies of old physicians. You're trying to decide, where you're going to live. You're going to live in central Pennsylvania or Mobile when you retire because you can't afford to live in a place like Durham. There are all these issues. The only good thing about getting old is that relationships become longer. Your friendship with people grow more and more over time. And that's I wouldn't say it offsets the whole dying thing, but it's pretty good. And Raj is one of those people I've known. Raj and I met interviewing for neurology residencies. That's how long ago it was. And, we were residents together, and we've been in contact for years, and both proud of each other's accomplishments. So I will let Wayne introduce Raj. I'll just throw up Raj's handsome face here, and Wayne can do the introduction.
Well, thanks, Rich. First, I also started with the thanks to Doctors Janice Massey and Wayne Massey for their leadership, but also generosity to set up as this and Macy's a lecturer, so we able to recognize leaders in neurology every year in the springtime. I'm going to be objective because, as you know, Rich know Rajiv for years. They did a residency together, so he might be biased. I actually only know Rajiv just in the last few years, so I'm going to be very objective. Dr. Rajiv Ratan attended an undergraduate and in Amherst College, then did an MD PhD at NYU School of Medicine. And then he actually moved to Chicago, Midwest, did an internship, and then moved back to East Coast, did a neurology residency in Johns Hopkins and a fellowship in neurorehabilitation medicine. He stayed at faculty for 2 years and moved to Harvard Medical School and become an attending consultant at the Bethesda Deaconess. And in 2003, he became the executive director at a Burke Neurology Institute and really one of the few freestanding research facility, has very dedicated program and in basic science, translation science, and clinical research. In the last 3 years at opportunity visit, I'm really amazed by the comprehensive and competitive program he built over there. So I was going through his CV and, here's all the metrics, numbers really just speak for him. He published 174 manuscripts, have been consistent funded by NIH since 1997. He's only a few people actually led 3 PO1 center grants and program project. The total NIH funding was over $18 million through his life, through his career. Not to mention, he actually led and formed a center of excellence in spinal cord injury in New York State with a $15 million grant from the state. He's also inaugural director for the multi-center trial platform to cure stroke or disability, and with a 5.5 million funds from the New York State. He holds 9 patents, made a many contribution to the science, and really, and including his a years of research just diving into the neuroprotective and ion chelator and protecting neuro from fatal apoptosis through the inhibition of a family of material enzyme, and also really made a remarkable and researching epigenetic protein as a target, how really for neuroprotection and repair in the central nervous system. Besides being a very productive researcher and leaders, he's also a great mentor. He trained over 30 postdoctoral fellows, and many of them become a faculty in neurology and neuroscience. And so let's welcome Dr. Rajiv Ratan, and his talk about his intermittent fasting, Alzheimer's disease, stroke, and neurorestoration, and a novel basic and a clinical perspective on treating disability. Let's welcome.
Thank you.
Wow, and that was a hell of an introduction there.
It was. Thank you very much, Wayne. Thanks for the invitation. It's been such a pleasure to be here with this guy and other friends. And I want to say to Dr. Massey that I feel particularly honored to give this lectureship, and I hope to follow an amazing scholarship, tradition of scholarship that you both have developed.
Thanks. And thank you, Wayne. Okay, Raj, all you.
All right. Let's see here. I just hit this.
Yeah, touch the screen, and then you can use the arrow sideways there.
All right. And the pointer is okay. Great. Well, I thought I would start with this old picture. This is a picture of our residency class as we were finishing up in 1992. And I think what you can see here, so I will say that Rich and I as chiefs had an amazing partnership caddying for this guy, Dick Johnson, who was the chair of neurology, and mentoring this group. It was really quite an amazing group. And And the things, you know, I think you all know Rich's amazing strengths. He has great taste, as evidenced by the fact that he's recruited many of you. And he is brilliant and funny. But I think one thing you also notice is he's actually aged much better than I have. I think the curves were actually on top of each other until about 2 years ago when he started taking Ozempic. And then our curves diverged. So I think there is a therapeutic lesson for me. I figured that. So I thought I would start with this picture of Rita Levi-Montalcini. As many of you know, she was a neurologist who won the Nobel Prize for her discovery of nerve growth factor. And she actually, I consider her, you know, we want to think of ourselves as coming from great lineages. And I'm a neurorehabilitationist. And so I consider her the first neurorehabilitationist because she developed and identified biological mechanisms that are very important for repair. But I also love this picture because it shows that winning a Nobel Prize does bring true happiness. So I'm gonna have two short vignettes. I've been told I'm on the clock, and I maybe I'll hope you'll have questions at the end. And the first is largely a preclinical story, which focuses on this concept of intermittent fasting and maybe gives you some insight into strategies that have been developed in my lab that we think have the opportunity potentially to impact neurological therapeutics. And then the second half, if I have time, I want to tell you about a novel clinical trial platform that we're trying to develop with Wayne's help. And so I think many of you know that therapeutics for sporadic CNS diseases, and probably familial too, have translated poorly from animals to humans. And there are many, many reasons why this is the case. But one of the reasons is that when we study animals, we tend to isolate. We use animals that are genetically identical, that don't have comorbidities, and often where we've kind of homogenized the process. And so what happens is we develop therapeutics that then once they get to humans don't work because there's this amazing heterogeneity in the way that disease is manifest. So, so if complex pathophysiologic events are happening concurrently, how are we going to target all of these pathways using a single drug? And you might say, well, we're not. And if you look at oncology, the path to success has been combinatorial therapies, but combinatorial therapies take an enormous amount of time. I'm going to offer potentially a solution how single targets can affect many pathways. And there's already evidence that this might be successful in the drugs that we use for neurological diseases. So this doesn't really reflect a novel idea. It's sort of just emphatically restating it. So the idea is to focus on this concept of homeostasis. And many of you know that in response to either physiologic or pathologic perturbations, what happens is we mobilize responses that allow us to reset an endpoint that is consistent with survival. And a good way to think about this, and these homeostatic loops are mediated through environmental sensors. So, and the example that I like to use, which all of you are familiar with, is when you have heating your own home. If you live in an old home, there are two pieces of metal that are separate. If temperature falls below a particular level, those two pieces of metal come together. It activates a heater that then heats the house and shuts off the thermostat. But it starts with a critical sensor. And so the question really is what are those sensors that exist in neurons or in the brain, and how are those sensors then activated to induce either post-transcriptional or transcriptional responses that ultimately feed back and deal with the stress. So you can imagine that in the body, we would have oxygen sensors or oxidative stress sensors, energy sensors, DNA sensors, amino acid sensors, all of these things that are critical for the survival of the brain. And when they go awry, you get the activation of these homeostatic loops. So I wanted to give you an example of a drug that's been developed that focuses on this pathway. And it turns out that the oxygen sensors include a family of enzymes called the HIF-prolyl hydroxylases. These enzymes are iron, oxygen, and 2-oxyglutarate dependent. And what they do is they take one molecule of oxygen and they stick it on this protein called HIF-1. And in all of our cells of our body, as we're sitting here, these enzymes are adding this hydroxyl group to HIF-1, which targets it for destabilization. So as we're sitting here, it's being synthesized and degraded. But if oxygen were to fall below a particular level, that enzyme would cease to function. HIF would not be hydroxylated, and therefore it wouldn't recruit the E3 ubiquitin ligase, the von Hippel-Lindau protein, and it would be stabilized. And so it then dimerizes with its partner, moves into the nucleus, and it activates all the genes that you would want to be upregulated if a cell were hypoxic. So erythropoietin, so you can make more red blood cells. VEGF, so you could actually make more blood vessels. Glycolytic enzymes, so you can convert energy metabolism from mitochondrial to glycolytic. It is a beautiful and elegant system that has evolved over millions of years and that actually garnered the Nobel Prize for Greg Semenza and Bill Kalin and Peter Radcliffe in 2019. Now, what's really cool about this is that now you have in a single enzyme, you have an oxygen sensor. So what you can do is you can develop drugs to those oxygen sensors, and you have a way of essentially turning on and turning off the adaptive response to hypoxia whenever you want. And for all of you who are clinicians, you know that one of the major circumstances in which this happens all the time is chronic kidney failure, where the body ceases to produce erythropoietin, people become anemic, and they become hypoxic in their tissues. And so it turns out that the liver is trying everything it can to synthesize EPO, and it can't. But if you use an inhibitor of these enzymes, now the liver will produce erythropoietin so that you don't have to give it systemically. What we've found is that we've developed drugs that target this, and it was highlighted in this Science Translational Medicine paper. We can actually treat a variety of types of stroke, a variety of eye diseases, as well as traumatic brain injury. So it's an example of how these homeostatic loops can be effective. So just I want this kind of sets up the data that I'm going to show you. So modulation of stress sensors by drugs allows the activation of resilience genes without inducing the stress itself. That's key. You don't want to have to induce hypoxia to induce this response. You could turn it on and off with a drug. These resilience genes, like for hypoxia, oxidative stress, bioenergetic dysfunction, have evolved over millions of years in multicellular organisms. So they are coordinated at a cellular, a local, and a systemic level. And what this gives you is it gives you a breadth so you can deal with the heterogeneity of disease mechanisms. They're deep because the organism needs them to survive. And they can be non-toxic if you intermittently activate them. So the persistence of activation of a stress response, which we see in all of the neurodegenerative diseases we look at, is problematic because the brain says, I'm activating the stress response, but I'm not compensating. And so what it starts doing is it starts deleting cells because it's trying to preserve nutrients. So I'm going to talk to you about some of our work on glucose sensors today. And many of you know, and this kind of relates to the title, glucose sensing, Alzheimer's disease, stroke, and neurorestoration. So many of you know that glucose is the brain is 2% of body weight, but takes 20% of the body's glucose. If you fast, as soon as you consume all the glycogen in your liver, you start to shift metabolism over into ketones. And you know, this is the basis for the ketogenic diet, which also has well-established evidence and support in the pharmacopoeia of neurology, used to treat epilepsy. And so most of the benefits of intermittent fasting are thought to relate to this process. You fast, you consume all your glycogen in your liver, you start to produce ketones. Once those ketones are produced, they have effects on the brain that are thought to be beneficial. And there's a beautiful review in the New England Journal of Medicine that was written by Mark Mattson, who used to be the head of neuroscience at, and the thought here is that, again, just to go over this, is that in your liver, once you consume all the glycogen, you have all these metabolic changes that start to consume fat. That's why it's thought to be good for weight loss. And then you have things that happen in the blood that affect many organs, including the brain. And in the brain, this equals changes in ATP, increases in mitochondrial biogenesis, improved performance and stress resistance, and most interestingly, increased cognition, probably through this protein called BDNF. So we have a lot of information about, you go through this intermittent fasting, you get all these benefits, but we have very little information on is how that actually occurs, what's in between. And the problem is this has been fodder for the charlatans, because they say some people are selling an intermittent fasting diet that's every other day, or 2 days out of 5 days, or 1 week out of 4 weeks. What's the right dose? And what people are saying is, well, we're using ketones as an evidence of target engagement, but it turns out that's not so effective. So the thought is that while you fast, you build up resistance, and then when you refeed, you take advantage of that resistance. So the question that we wanted to address is, how is low glucose during fasting sensed? Again, getting back to my analogy about sensors, to be able to confer benefit on the brain. And again, this is so critical if we're going to think about moving this into therapeutics. So the tool that we used is a tool that's probably familiar to many of you. It's an analog of glucose called 2-deoxyglucose. And just to take you back to the horrors of high school biochemistry, you'll remember that glucose comes into the cell and it undergoes glycolysis that leads to pyruvate, which then goes into the mitochondrial electron transfer chain to generate ATP efficiently. It can also be used in the hexose monophosphate shunt to generate reducing equivalents. And this is how you combat oxidative stress. So glucose is very important. And you might say, well, Raj, why would you use 2-deoxyglucose? Well, it's sort of like Ozempic, isn't that right? You want to be able to eat all the good things, and particularly if you come from my culture where we love sweets, you want to be able to eat all the good things but have all the benefits. And so the lifestyle advantages of 2-deoxyglucose are enormous. So these are pictures of glucose, and it just shows that 2-deoxyglucose differs from glucose in one hydroxyl group. It's very, very similar. And what that means is that when glucose is taken up through a glucose transport, 2DG, it is metabolized by this first enzyme, hexokinase, but the product feedback inhibits this, so glycolysis is inhibited. And this is why 2DG inhibits the ability of the cell to actually use glucose. So we were interested in saying, okay, we can use 2DG as a tool to modulate glucose in neurons. How are we going to study cognition in a dish? We're going to start out with very simple systems. And we said, okay, let's use brain-derived neurotrophic factor as a proxy for its behavior as a proxy for the benefits of 2DG. And let's ask whether 2DG increases or decreases BDNF. And you know, BDNF, decreases in BDNF have been linked to aging, Alzheimer's disease, and chronic stress. There are polymorphisms in BDNF that actually are linked with better recovery from stroke and traumatic brain injury. And a lot of the benefits of these interventions, exercise, enriched environment, and antidepressants, are thought to be mediated through BDNF. So again, what we do in my lab is we start with very simple systems along the theoretical axis of reductionism, and then we move to progressively more complex systems. So if you simply take neurons in culture, and these are neurons in culture along the bottom, stained with the live cell stain, and you can see you can drive 2DG levels up pretty high, 15 millimolar, and not have any effect on the viability of the cells. But what is fascinating, and I would just have you focus on A, is that adding 2DG dose-dependently increases BDNF. This really surprised us. So this suggests that decreasing glucose actually enhances the synthesis of a gene that's involved in plasticity. And it starts to be vaguely reminiscent of what happens during intermittent fasting. And this just shows that you get an increase in protein. So this was so heretical in our view that we had to do a whole series of controls where we looked at human neurons with Guili Ming at Penn. We got the same result. We actually looked in vivo in mice, and we found that using BDNF as a proxy, that 2DG could upregulate BDNF, but the concentrations of 2DG that were required were 10 to 20-fold less than what people had used previously to induce the benefits of intermittent fasting. We then took mice which had heterozygous deletion of the transporter for glucose in neurons. And we asked, is BDNF upregulated? And it was. So in all these circumstances, reductions in glucose metabolism were leading to an increase in this protein that's involved in plasticity in the brain. So because the concentrations that we used were 10-fold less, or 10 to 20-fold less than what had been previously used, and we didn't see any ketosis, before we did any mechanistic studies, we wanted to be sure that there was potential therapeutic benefit. So what do is we use diseases in mice as a proxy for looking at the integrated benefit of an intervention. And so by using BDNF as evidence of a dose or target engagement, and using things like behavior and long-term potentiation as integrated measures of neuronal benefit, we looked at what happens. And I'm just going to go through this really quickly. I don't want you to get, the data is busy. What I will tell you is whether you look at an ischemic stroke model or a hemorrhagic stroke model, you see benefits in behavior. Those benefits, so 2DG is started 1 day after the stroke, and that we don't really have any drugs for 24 hours after stroke. So, and yet you don't see the benefit for 2 weeks. And that suggests that probably the brain has to synthesize something that's enhancing this repair. We also looked at an Alzheimer's model. This is what's called the 5X FAD model. It has 5 Alzheimer's linked mutations. It's a very aggressive model that Rich would tell you probably has very little to do with the human condition, but it is a proxy for whether this can have an intervention. And again, it's a busy slide. Let me just point out a few things. One is that if you look in B, so what we did in this case is we actually used a pump to deliver 2DG directly in the brain to really remove any doubt that this could be a direct brain effect. And I think the clearest, you see here, if you look at BDNF levels, they go down in the 5X FAD mouse, but if you treat with 2DG, they're normalized. And that behaviors that are used to test short-term memory or long-term memory, all normalized in the presence of 2DG directly into the brain. So this gave us confidence that, in fact, 2DG the doses we are looking at directly in the brain could have benefits, and that this was associated with increases in BDNF. So what's the mechanism? And I think this is what we think is exciting and has the opportunity to be transformative. So the first thing you would say is, well, Raj, how do you know this isn't due to changes in ATP? ATP activates AMP kinase. There are drugs that are developed AMP kinase. You manipulate AMP kinase, no effect. The effect was dependent on new transcription. So again, getting back to my original model of building resilience, 2DG is inducing genes that are somehow increasing BDNF and other things. And we thought, oh, it's having such broad effects in multiple models. This couldn't be an effect on a single gene. So what we did is we did what's called RNA sequencing. And many of you probably are familiar with this technique, but if you're not, you basically take all the RNAs, you convert them into cDNAs that are more stable, and you sequence them. And so you can get a read on all the RNAs. And for us, this was really an important experiment because we did it with Dan Geschwind at UCLA. And so it was a way of us sort of taking results from our lab and verifying that they actually were true in another lab, which I think is really important. So we got an unbiased view of all the RNAs. And so this, if you focus on this graph here, blue is low levels, red is high levels, blue is control, pink is 2DG at 1 millimolar, red is 2DG at 10 millimolar. You can see there's a beautiful dose-dependent increase in BDNF, but all these other genes are also known to be involved in plasticity. So it's not just one gene, it's a cassette of genes. And what you can do, what's really exciting about being able to do this type of analysis is you can actually develop gene ontology groups. You can see, oh, there's a program that's activated. And it turned out that the number one most activated program involved this response called the unfolded protein response. And if you look at where in the cell this was happening, the leading gene ontology groups were this oligosaccharide transfer complex, the endoplasmic reticulum, and the Golgi. So you might say, and we said the same thing, was like, what does that mean? And so, but I think it's important to try to understand it, what the unfolded protein response is. And if you focus over here, so just taking you back to cell biology, remember the endoplasmic reticulum is where your proteins are, many of your proteins are synthesized, isn't that right? And what happens is if they're not synthesized, and this is thought to be the case in many neurodegenerative diseases, if they're not synthesized correctly or folded correctly, it activates a very elegant adaptive response called the unfolded protein response. And it has 3 arms. And I won't go through those arms, but those arms do all the things you would want to do if you had unfolded proteins. You want to stop making new proteins. You want to increase chaperones that would allow you to refold proteins. And you want to degrade the proteins that are not folded properly. Well, the way and this oligosaccharide transferase complex, it's actually quite simple. So in eukaryotes, what happens, we use sugars, mannose and glucose, and we stick them onto the end of our proteins to make sure that they're folded properly. So if you don't have enough glucose or mannose, these proteins, these green and blue marks, these proteins are not folded properly. It triggers this unfolded protein response, and then this unfolded protein response has the opportunity to activate genes. What was really surprising is that these genes involve plasticity genes, and we'll talk about that in a second. But just so you can remember this, glycolysis normally generates pyruvate for the mitochondria. These N-link glycosylation, now this happens, 50 to 60% of our proteins are N-link glycosylated. And so you can imagine when you fast, so many of those proteins that were glycosylated are not going to be. You're going to activate the stress response. You're going to upregulate not only genes that are involved in plasticity, but also proteostasis. And that's largely as a result of this, also this effect on mannose. And the reason I show that is that you can actually, if you load neurons with glucose or mannose, and again, you can just, I'll just tell you, then you can abrogate the increase in BDNF that you see in this. So this is the model, and I promise Rich no more gels. The model is that when you have 2DG, you decrease glucose and mannose, you have decreased end-link glycosylation. That activates this unfolded protein response. It paradoxically translates a transcription factor called ATF4. ATF4 is like a lock. It's like a key in a lock. It goes into the nucleus, it binds the DNA, and it turns on all these genes. And what was really fascinating, and again, is that we can actually probe whether ATF4 is bound to all those genes. And I will tell you that it is, and it actually was induced in all, you can do these volcano plots that show kind of correlation between statistical significance and levels of induction. All the plasticity genes are actually upregulated. So end-link glycosylation is actually a glucose sensor and a mediator of adaptation to low glucose. And we published this in Neuron. And the reason we think that this is such an exciting new pathway is that because it really gives a whole series of potential biomarkers that now allow you to think about how you would apply intermittent fasting to humans. You can look in the blood and see if ER stress is activated. It also suggests that 2-deoxyglucose and maybe better versions of it would be better therapeutics than actually intermittent fasting. So, so the idea is that when you go to sleep at night, and this is the reason to eat at 6 o'clock and not after 6 o'clock, when you go to sleep at night, you fast. You probably generate a level of glucose deprivation that inhibits end-link glycosylation, that upregulates genes that are involved in plasticity and proteostasis, and that protects you against a variety of different diseases and also potentially enhances your recovery. So the idea is that when you sleep, you're kind of fixing the hardware, and then when you wake, you refeed, you translate all those proteins, and everything is better. Now, why would this develop? You would say, well, why would you increase cognition when you're starving? And the reason is because probably there was selection of, if you were starving, you had to think strategically about how you were going to find food. So there was a evolutionary pressure to find ways to be more creative and think better when you're actually starving. So, and one of the other things that's important is that the concentration of glucose in our blood is 5 to 6 millimolar. The concentration of mannose in our blood is 300 micromolar. So end-link glycosylation is much more sensitively affected, potentially by drugs, than energy metabolism. All right, so that's the first part of the talk. How am I doing on time? I have 10 minutes, is that right? For this last part? Okay, 12 minutes, yeah, okay. All right, so I'm gonna tell you a little bit about so our institute is focused on brain repair, and I think we would all agree that, you know, chronic neurological disability is reaching epidemic proportions, and we need a practical strategy to how to understand to repair the brain in humans. And I think what we're part of a movement is to help all of you understand that traditional rehabilitation treats disability. We, as neurologists, really need to be focusing, treats disability at the level of disability. We, as neurologists, need to be thinking about treating disability at the level of repairing the brain. And I think this, so the idea is that if you have a stroke, I think all of you know, the right side of your brain is, the right side of your body is weak. As a result of that weakness, you won't be able to get across the room. Current rehabilitation puts you in a wheelchair to allow you to get across the room, or teaches you how to use the good part of your body. If you work behind a desk like I do, that's fine. If you sell popcorn at Yankee Stadium, you're not going back to work. So we really, I think the opportunity for us as in neurology departments is to figure out, how do we get arms and legs moving again? And right now there is exciting evidence, I think you've probably heard of this, that you can now get arms and legs moving years after paralysis beautiful work from Gregoire Courtine at the EPL in Lausanne showing that people can actually walk again after being paralyzed for years from spinal cord injury. Anecdotal evidence from the stroke group at Stanford is injecting stem cells into the peri-infarct region. And I think, as Wayne knows, they have a subset of patients the day after they get injected, they're completely normal. So these are proof of concepts that there are physiologic breaks on repair. So, but the brain is unlike the kidney and the liver. We can't just fix the hardware. You have to reboot the software, isn't that right? So the way that we build skills, those of you who have had kids or nieces and nephews, you know, the child brain is plastic, but that plasticity isn't converted into skills without it being shaped by the environment. And so you would agree, and I think Wayne's already testing this in his current Transport 2 study, that any therapeutic for the brain can't simply involve throwing an antibody at it. You have to likely combine that with some strategy to reacquire skills. And the problem is that right now, we can standardize this part of it, the antibody part of it. We haven't standardized the training part of it very well. So what we decided to do is we said, okay, can we build a chronic clinical trial platform that takes advantage of robots that have been developed at Berkeley in collaboration with MIT? And what these robots do, you can see somebody, this is a patient with stroke who has their left arm in the device. And they move the device from a center circle to concentrically organized circles. And what has been figured out over the last 15 years is that doing this every other day for 6 weeks gives you the greatest impairment reduction. And so this becomes a standardized way to get a small improvement in impairment in the chronic phase. What's also nice about these robots is that you can actually take the data and send it through the cloud to a centralized data coordinating center. So you could have many sites doing trials and have all of it coordinated at a central site. You don't need humans because you can do kinematic outcome measures with these robots. And that takes the human component away from actually doing the evaluation. And it's been supported in a number of different papers. This paper from the New England Journal of Medicine in 2010. So it's an intervention that has a lot of history. But even though robot-assisted therapy is clinically meaningful, the changes are really small. And what people have been trying to do, and I think Wayne would agree with this, they've been trying to squeeze as much disability and handicap benefit out of a small change in impairment instead of figuring out how do we get a large change in impairment. And that's what we want to do. We want to say, what's the secret sauce for actually getting arms and legs moving again? And so we know that if we pursue this, that we can succeed, but there's going to be a lot of failure along the way. And so the thought was to put together a platform trial. So as many of you know, the idea of one drug, one trial is kind of becoming passé because we spend all this money to build an infrastructure for doing clinical research on a single problem. And then after we're done with one trial, we deconstruct it. Why not just keep it going and then keep adding more interventions? They're doing this at the Healy Center in Boston for ALS. We wanted to do the same for chronic stroke. And the idea is, can we find an intervention that changes the plasticity of the brain, like a drug or a biological, a stem cell or an electroceutical, that when combined with this standardized robotic training, gives us really large changes in impairment. And that's the overall goal. And so this is kind of the idealized vision of it, that you would have this training. You would be testing multiple drugs at the same time. One would potentially give you larger benefit. That would become then the standard of care. You would have to use fewer control groups. That's the vision. And we've done this, at least begun to set this up. And I will tell you, this has been the most challenging exercise of my professional life. It's been incredibly frustrating. We have 4 organizations that have come together, and the problem, our original, the state of New York funded this, but the problem that we had was that we said we'll build this structure and then tell companies to come and leverage it. They don't have to pay for it; they just have to use it. And there are no companies interested in chronic stroke, or the companies that are interested in chronic stroke, they're worried, oh, well, if we take our drug and we combine it with the robots, does that mean we're only going to get an FDA indication with the robots? What if there's a negative interaction? There are companies that don't want a go, no-go decision at this point. And so I think we've decided that we really have to do this as an academic effort. And our initial hypothesis to be tested is an exciting one. I just wrote an R01 on this, and so I'm really excited about it. And the idea is that chronic stroke disability persists because it's like the brain is undergoing persistent autoimmunity. So there's a really exciting new pathway called the CGAS-STING pathway. This is a DNA sensing pathway. It activates microglia, and we think it's persistent because there's an enzyme called Trex that gets modified to make it persistent. And then the activation of this pathway leads to the upregulation of these cytokines called CCL3, CCL4, and CCL5. They then activate a receptor called CCR5 that closes the window of plasticity. And you may be aware of beautiful work from Alcino Silva and Tom Carmichael showed that CCR5 deletion is effective after stroke. And it turns out there's an FDA-approved drug called Neurorestoration that was built to inhibit the HIV. So CCR5 turns out to be the HIV receptor. And so Neurorestoration can inhibit this receptor, open the window of plasticity, and induce remarkable improvement. So we're going to test, since it's FDA approved and available and safe, we're going to test that in combination with the robots. And so, and this is just some of the outlines but I will tell you, this has been incredibly expensive, incredibly frustrating, and incredibly difficult. And I hope that we could find a way to collaborate with this department to kind of move this forward, because I think it needs to happen, and the field needs it to happen. So I thought I would end with this quote from Levi Montalcini. And actually, I put this quote here because I totally disagree with it. I actually think that right now our understanding of how stress leads to resilience is greater than it's ever been. And we know from epidemiological studies that, you know, exercising regularly, eating the right things, having a good social system, all those things epidemiologically equip you. And I think what they're doing is they're activating these stress response mechanisms and building resilience. And so I think it is actually perfection that's written to the program, written into the formidably complex engine that is the human brain. And I obviously wouldn't have been able to do any of this without collaborators. You know, Wayne has graciously helped us. We'll hope to get more involved, and then the work in my lab over many years from many people. So with that, I'll stop.
Wow, Raj, that was fantastic and right on time. All right, so questions from the room and from the internet. Joe?
Anything bad happened?
Right, so at this point, we have not seen any obvious toxicity, but I think your question highlights the fact that we usually look at a small subset of behaviors. We should be looking more broadly, but at this point, we haven't seen any adverse. And I think it has to do with this fact that we're using a really low dose. So currently, 2DG is being used to treat cancer, and the dose that's being used in humans is 60 times higher than what the dose we would predict would be. This is really taking advantage this is really leveraging the fact that 2-deoxyglucose turns out to be 2-deoxymannose, and it can inhibit that end-line glycosylation very sensitive. To follow that up. So we're talking. So we're talking right now with people who've done the most in cancer, and they're actually doing studies in India. So it's a great question. I think our I think it's a great idea. Our priority turns out that there's a fluorodeoxymannose more selectively affects the glycosylation pathway versus glycolysis. We think that's actually the drug we would ultimately take in. So we're trying to get funding from foundations first to evaluate different analogs and start to decide whether knocking down part of the end glycosylation pathway might be a better target. So I think those are things that we're doing.
Wayne, let's do a quickie. Raj, how does your dose compare to what Tom Satula's investigating for 2-deoxyglucose in epilepsy?
Yeah, so that's a great question. I think that the doses he's using are much higher, like 10-fold higher. And what we see, it's a really good question. What we see is that as we drive up in a dish, or actually in vivo, as we drive up the concentration of 2DG, we actually see BDNF is inhibited. And we think that happens probably because you're starting to affect those energy metabolism pathways and you're inhibiting translation.
So we'll come to Wayne right after this. So Dr. Shah says he wants to know, are you doing any intermittent fasting? And I would throw into that, do you think Ozempic does some of the things you're talking about?
I think I'm not doing intermittent fasting, although I think from looking at me, you should say I should. And I think it's because I don't believe that we have a biomarker yet. We think we have one in mind, but don't have an established biomarker but I actually think Ozempic is doing a lot of things. So Ozempic, as many of you know, has many different functions that essentially tell the body it's full, so we eat less. It improves insulin function. It affects the transit of nutrients through the gut. All of those things should reduce glucose. And I would predict, so it's a great question, Rich. I thought about it after our dinner last night that we should look at whether Ozempic turns on.
There was no intermittent fasting going on at dinner last night.
That's right. That was the full-on anti-intermittent. But I think it would be interesting for us to look at whether Ozempic upregulates BDNF and all of these protective genes in the brain. It's a great.
Wayne?
So. Based on what I see, right. So I think again, it highlights the fact that it's a great idea. There's a lot of work to do. So interestingly, in hemorrhagic stroke, 2DG actually protects the brain. So if we were to give it earlier, we think we would actually block death. So I think the question always is once you have a biomarker that tells you what's the therapeutic, when you've engaged the target in a way that's safe and effective, then all these questions become readily testable, right? But until then, we wouldn't know, are we helping people? Are we hurting them? So I think it's a great question. They may be already naturally doing it, but remember, with fasting, it may be that because glucose can be converted into mannose, you probably have to fast for a lot longer to have the effect we look at, as opposed to using 2DG or FD mannose. So that would be, I think, again, I hate to say it, it's not quite ready for prime. It's getting there.
One last question. Yeah.
Yeah. Well, in my lab, I think one of the things we try to do all the time is have, before we do make the next step, we always have it reproduced by another lab, and we haven't done that yet. We usually try to do it before the first publication, and I will tell you, there have been some circumstances where things don't replicate. So I would like to see this reproduced in another lab, and once that happens, then I think we should, with these appropriate analogs, we should go full bore on trying those things in primates.
So, you think, I mean, benefit maybe from the, like, if you take some time, do you think on the functional level, like a Microsoft level, it changes the functional brain connectivity?
I think so, and I really, I mean, I think in our institute, I think we've invested heavily in faculty who study circuits, and I think circuit repair is the future, because there's probably more in common among diseases that involve the same circuit than a single disease with multiple circuits. So I think that is the future, and the technology is around me to be able to address those.
Raja, I had said that was the last question, but Sina has an interesting final question about in the ICU, continuous feeding versus bolus feeding.
Yeah, those are great questions. I would love to know the answer, and if you have access to the data that could address it, I would be very interested in the answer, but great question.
Again, Raj, just fantastic job. And Wayne Massey, thank you for getting involved. Wayne Feng, thank you for inviting Raj. Joel, great talk too, and everyone have a great day.